And so what we're really looking at is the plumbing as opposed to the electricity in the brain. That's a gross oversimplification, but it's a nice heuristic for thinking about it. Now, one important property of the bulge response is that it's very, very slow in relation to the brain activity. So here's a very famous figure in the middle from Lomothetis and colleagues. And they were able to show in 2001 for the first time that this thing that we measure in MRI called the bulge response is actually directly related to the firing of brain cells. This was an important demonstration. It's surprising to me that it hadn't happened 10 years earlier when MRI was really coming into the forefront, but this, to my mind, is really the first definitive demonstration that that was the case. And they were able to measure simultaneously SDF, which is the spike density function, so the number of firings per minute, or per second probably, of a brain cell, and multi-unit activity, which roughly measures pretty much the same thing, the local field potential, which is the change in electrical charge or electrical activity in a tightly defined area, and also the bulge response, that's the blood oxygen level dependence. And what we can see is that the spiking, that is the actual firing of the brain cells, happens pretty fast. So within a couple of seconds, you present a stimulus for, say, 4 seconds, and fairly soon afterwards, the spiking comes back down again. So that's pretty clear from the green and the blue and the black lines there. But the bulge response, which is the thing that we're trying to measure, takes much longer. In fact, it takes about 6 seconds to reach its peak, and it takes probably between 16 and 32 seconds to go all the way back down to baseline again. So what we're really trying to measure in MRI is this big hump of pink activity in the middle. And you can imagine that requires some fairly fancy statistics in order to be able to do it properly. So that's the first thing that we're measuring in MRI that's important to understand. We're really measuring the blood oxygen level dependent response. And so anything that we make an inference on has to bear that in mind. It's the hemodynamic response, not the electrical activity exactly. So what do we get out of the MRI machine? So let's say we put our subject in the scanner, and we have given them a particular task to do while they're lying in there, and we want to try and estimate what the differences in brain activity are using this indirect measure, the hemodynamic response, or the blood oxygen level dependent, over the course of time. So let's say we give them a working memory task, for example. They have to keep something online in their head, so they don't have to remember a phone number, which is only 6 digits, so they might say, oh, 333-987, 333-987 over and over again. And then let's say we give them another trial where they've got to remember 10 digits, so we manipulate the difficulty of what they have to keep online. But what we actually get out of the scanner is something that looks roughly like this. So many hundreds of images in most cases, normally one every 1 to 6 seconds or so, that represent slice by slice, so normally we collect this in individual slices at a time, going up or down the brain, and that can be reconstructed to give us three-dimensional images of the brain, particularly the brain's blood oxygen level dependent signal. So you can see the first thing is they're quite blurry. So on the bottom left there, we've got a structural scan. So that's the scan with the very nice resolution, so 1 by 1 by 1 millimeter. So normally our functional scans are about 1/3 or maybe 1/4 as accurate in terms of their spatial localization. And they contain, nevertheless, several thousand voxels. Remember, that's the volumetric pixels. So several thousand 3 by 3 by 3 millimeter boxes, each of which represents one measurement. So we have many, many measurements. And the reason they're blurry is because what we're measuring is blurry, actually. So the bulge response itself happens to be quite blurry. So there's not much we can do to get the resolution that much better, because the blood flow isn't so well localized as the electrical activity itself. So then what do? Well, we do a process called pre-processing. So we have to make sure all of these scans are nicely lined up with each other. So we have to perform some algorithms in order to correct for the fact the subject moves ever so slightly. So they move their head up and down or back and forward during the scan, which is a problem. And so on the bottom left, you can see a diagram of movement correction. So this is a subject who moved very little. The y-axis there is in millimeters. So they managed to move over 200 scans, which is probably somewhere in the region of about 8 or 10 minutes, only 1 millimeter in either direction, so this is a good-looking subject. But we nevertheless do some correction to put all these images back into exactly the same space. In some cases, we correct for the fact that we've taken the slices at different times. Remember, we take one slice at a time, which typically will take between 50 and 100 milliseconds per go. And then we do a process called spatial normalization. We want to try and put all of the brain images into a common space. This is a bit difficult to think about. So one way to think about it is that my brain and your brain and everyone else's brain are ever so slightly different shapes. But there is an average of all of our brains, which is a characteristic shape. And so there are various centres, one is the Montreal Neurological Institute, that have created these average brains of many hundreds of individuals. And so typically we warp everyone's brain, so we squash it or squeeze it a little bit, or stretch it, so that the brains all look like the same kind of thing. We can't do that perfectly, unfortunately, but we can have a pretty good go at doing it. So you can see the top left is actually this template image, the thing that we're trying to make all the brains look like. And then this is the brain images of the subject that I chose to give you the example of. So you can see, by and large, they match fairly well to the template that we're trying to match it to. And then what we do, because every subject is still a little bit different, is to actually blur the data even more. So we started off with blurry data, and then we make it even worse. So then we blur it so that the different areas of the different subjects that are ever so slightly different aren't quite so different as, anymore. So we normally actually get to about 8 millimeters resolution as opposed to the 3 millimeters that we measured in the first place.